Hi, good evening. I think it's just turned 6:30, so I think we should start. It's already late in the afternoon. So welcome everyone. Welcome for joining this evening. It's a rainy night in Potchefstroom, so I hope if you're a local attending, you have a pancake and a coffee next to you. Tonight's speaker is Professor Chris Vorster. He's a bit of a celeb in rare diseases in South Africa, so I'm not going to introduce him all that much. So Prof Vorster is the chemical pathologist at the Center for Human Metabolomics, and he has also recently been appointed as the director of the NUE program. And this program is specifically directed towards diagnosing undiagnosed diseases. I'm not going to say too much because that's what this talk is about. Thank you for joining. I see we're about 20 participants currently, so I think, Chris, it's safe that you can start. We will have a few questions and answers afterwards. And then Chris just asked that he's going to ask a question or ask a reaction from you. And I think if you can then just click on that raise hand button, we will appreciate that just to see if we can get a reaction from the audience. Thank you. Talu raised his hand so we can see that it's working. Right, okay. Thank you. Good luck, Chris. Enjoy it.
Thank you, Ilse. I couldn't actually see the raise hand. I don't know if people can also do the thumbs up thing.
If you click on participants there, Chris,
okay.
at the bottom, and then you'll see the raised hands next to, there's no raised hand currently.
Okay,
there is. Ansi is raising a hand. Did you see?
Okay, fantastic.
Okay. Right.
Yeah, I'll see if I can work with that. Thank you very much, Ilse, and thank you to everyone who is here in attendance. I'm going to talk a bit about our proposed approach for rare undiagnosed disease patients, and in particular the UNWE program. We launched the UNWE program at the RareX conference in February, and I am going to repeat some of the slides. I must say the session was very poorly attended by clinicians, so I hope to get to you now. For those that did attend the session, I do have some new slides as well, and I hope you will find that of value. Okay, so to get started, there we go. Okay, so a while ago, we were fortunate enough to do a short pilot project in South Africa where we did whole exome sequencing. The sequencing was done in South Africa in a number of patients. We had to recruit the patients in record time. They were not that well selected, but I'm quite surprised at what we have found in the pilot project. And in one of the patients, we detected a variant in the cell gene. So we're going to approach this case study in the way in which we do it in the laboratory, which typically we don't have clinical information and so on. And I'm just going to take you through this journey of this particular patient. Now, I've listed two variants here. The one is from the patient, and the other one is a variant that I found in literature. And at this stage, I'm not going to tell you which one is that of the patient. And the question tonight is, are both of these variants pathogenic? Is just one pathogenic? And if it's just one, which one is it? Or are both of them benign? And if you're not familiar with this this annotation, then let me just take it through you. So we've got the transcript number here, then the name of the gene, which is cell, then the cDNA position of the variant, which is at position 2007. And you can see that there's a duplication there, and that duplication results in a frameshift mutation that then leads to truncation of that protein a few amino acids later. And that's normally the case when there are bases that are duplicated or deleted that's not in multiples of 3. So this is what we would call a null variant or a missense variant or premature truncation. And often it's associated with premature degradation of the protein and basically a loss of function. So in this first variant, you can see that this, like I said, is in position 2007. The patient is heterozygous for this variant. In other words, the variant is present in only one allele. And henceforth, I'm going to refer to this variant as V670. The other variant that I want to present is, of course, also in the cell gene. In this particular instance, it's a deletion at position 147 and 148, which then also results in a premature truncation of the protein. This patient is homozygous for this variant, so both of the alleles are affected. And henceforth, I'm going to refer to this variant as F50, just to shorten it a little bit. So is there anybody at this stage that would like to tell me whether these variants are pathogenic or not? If so, then please raise your hand. Hopefully not too many people are raising their hands at this stage. Okay, so hopefully what you want to ask me now is, or what you want to say is this is not really fair, because I don't even know what the cell gene is and what it does. And if that's the case, you're in good company, because I didn't know what it does either. So let's just get into the gene a little bit more. So there is a OMIM entry for this gene, and you can see it's located in the long arm of chromosome 9. It's a 10 kilobase gene, so it's not that big, and consists of 11 exons. And then you can see here that the F50 variant is located in exon 2, and the V670 variant is located in exon 11, the very last exon of the gene. The gene encodes for a protein carboxyl ester lipase, which we don't work with in inherited metabolic diseases, but of course I can deduct what the function of this protein would be. And OMIM also gives a description. So basically it says that it's produced by the mammary gland as well as the pancreas. It was previously called bile salt stimulated lipase, which hints to its way in which it works. So it is present in the gastrointestinal system, or secreted in the gastrointestinal system, and then in the presence of bile salts, it can then do its function, which is obviously to hydrolyze lipids. So if we go into this a little bit more in depth, so it's a major component of pancreatic juice, like I mentioned, hydrolysis of cholesterol and other lipids, and then secreted in the duodenum, and apparently very important for the digestion of milk fat in newborn infants. So now that we have an idea of what this gene is, what it codes for, and what the protein function is, we can get a better feel of, the meaning of this particular variant. And we get a feel that it may actually be of clinical significance. So in the laboratory, we don't have clinical information. So the next step perhaps is to look at the ACMG classification of this variant. And the classification has become, I wouldn't say complex, but it's become quite laborious. And so, we tend to make use of automated classification engines, and that has its advantages and disadvantages. And in this particular instance, I used Franklin to classify these two variants. And you can see that Franklin classified them both as likely pathogenic. And it's based on the criteria that it's a null variant or a missense variant or a premature truncation of the protein. And like I already mentioned, is that these variants typically cause a complete loss of function of the gene. So it's such a variant in a gene where a loss of function is known to be a mechanism of disease. So that's a pretty strong criteria for being pathogenic. And then also the available population data that we have indicates that this variant is present at very, very low frequency in the population, which hints to the fact that this is likely to be a rare disease. Okay, so we've got the two variants. We've got the ACMG classification. I didn't show all the criteria. I just showed the ones that is really of relevance to this case. So now perhaps at some stage, we may want to consider, what is the disease that is associated with this gene? And again, I am making use of Franklin here. And you can see that it is associated with maturity onset diabetes of the young type 8. So I never knew that there were so many types of MODY, but apparently there are. And we get some additional information here, which is extremely valuable. So the onset of the disease is a young adult onset. So this is typically, in Franklin, this is between ages 15 and 40. The mode of inheritance is autosomal dominant, so that's also quite important for our particular case study. And then the clinical features that's associated with this is abdominal pain, abnormality of the exocrine pancreas, and really pancreatic insufficiency. And I can also perhaps say that eventually acute and or chronic pancreatitis, hyperglycemia leading to maturity onset diabetes of the young, and then of course also elevated biochemical markers. So that's the clinical phenotypes that's associated with the patient. Now, up to today, I've actually not received any clinical information on this patient, so I decided to make some up. And I hope this is fairly realistic. So the patient is 18 years old, and it is a male patient. And the clinical information that I'm going to give you tonight is that this patient is presenting with recurrent abdominal pain, impaired fasting glycemia, and intermittently raised AST. So I think you would agree with me that we have a gene, we have a variant that's classified as pathogenic. We have an associated disease. The disease is the inheritance model fit, and our clinical presentation fit as well. So at this stage, we can conclude that both V670 and F50 are pathogenic variants that cause maturity onset diabetes of the young type 8. V50 is homozygous, so we'd probably expect this patient to be much more affected than V670, which is heterozygous. So this is similar to the situation with familial hypercholesterolemia, where if you're heterozygote, you're mildly affected, and if you're homozygote, you are severely affected. So at this point, if you agree with me, perhaps you can raise your hand, and I'm going to see if I can actually see who's raising the hands. So there's a couple of people that are raising their hands. And the others, I'm not sure if you're not agreeing or if you are in need of more information. Oh, there's some more people now that are starting to raise their hands. Okay, so thank you very much for that. Now, the one thing that is a bit of a concern to me here is not everything adds up. Okay, so I just want to try and see if I can. There's a part of the screen that's now blocked from my side. Okay, I think I'll be okay. So not everything actually adds up here. So the first thing is that this is an enzyme that causes hydrolysis of some lipids. Okay, and so what I would expect the clinical presentation to be is fat malabsorption and then vitamin A, D, E, and K deficiency. This is what you would typically see in a patient that suffers from fat malabsorption. And the other thing is that just working with a lot of enzymes, we know that normally when you have an enzyme deficiency, the inheritance pattern is either autosomal recessive, so both alleles need to be effective, or it's X-linked. It's very, very seldom that it's autosomal dominant. In fact, I can't think of one example in inherited metabolic diseases where a disease is, where an enzyme deficiency is inherited in an autosomal dominant way. And if you then compare this to the MODY 8 presentation, which is intermittent abdominal pain, diarrhea, not steatorrhea, but diarrhea, diabetes and insulin resistance, suggesting endocrine pancreatic dysfunction, not exocrine, which is what we would expect with when there's an exocrine enzyme that's deficient. Also general exocrine pancreatic dysfunction that probably explains the diarrhea as opposed to selected, which should have presented with steatorrhea. And then the acute and chronic or acute and or chronic pancreatitis, which I think it's hard to explain given the fact that this is simply a deficiency of one enzyme that causes fat malabsorption. And then even what I didn't tell you is, but if you go into the imaging of these pancreases, they're abnormal. So there are some things here that doesn't actually quite add up to what I suggest that the diagnosis should be. So if we think back to the ACMG criteria, a lot of this relies on the idea that this is a loss of function in a gene where a loss of function is a known mechanism of disease. So the first thing that I would like to just mention here is that, perhaps we should check that fact. Is it really a case that a loss of function in cell is associated with disease? So I had a look in the literature and I could find one publication where a mouse knockout model was established. And the main findings in this publication was that the cell knockout mice at normal growth, normal development, normal glucose metabolism, some of the female mice did show slight abnormalities when fed a high-fat diet, but nothing statistically significant. So the conclusion of these authors were that the cell modi phenotype could not be reproduced, and that a cell loss of function probably does not cause disease. Okay, so how do we bring this together with what we see in our patient? The clinical presentation of our patient seems to fit, yet we now have some evidence to say that loss of function in cell doesn't actually cause disease. So is this, do we have the wrong variant here? Okay, so some reading helped. So probably at this stage, we need to do some more reading, and some more, and some more, and some more, and some more, and some more. Okay, and finally, after going through all the literature, this is what is at the heart of the matter. So the cell gene, and in particular exon 11, has got a variable number of tandem repeats that it probably acquires, or most likely acquires, from a serodeficient, or from a serogene. Now, why this is important is that this VNTR region is rich in O-glycosylation sites. So, after the protein is translated, there's post-translation modification, in particular O-glycosylation. And as we know from second-year biochemistry, that alters the 3D configuration of the protein and it may change its function. So in this particular instance, if you do have a loss of function variant in exon 11, where you do lose those glycosylation sites, the protein is not degraded, but in fact, what is expressed is a protein that tends to form oligomers that is quite stable. And these oligomers have been shown to be cytotoxic to the pancreas, and then results in exocrine and endocrine pancreatic dysfunction. So very interestingly, here's some information from ClinVar. It's not the best, but what I actually wanted to show you, I couldn't get permission. It was copyright protected. But this actually does show it to some extent. So here's all the variants that has been described in the cell gene. And as you can see, is that most of them are lying here in the VNTR region. There are two likely pathogenic variants here. I'm not convinced that they are pathogenic, and ClinVar is full of mistakes. So it's just something that you've got to be careful with. And then if I filter for null variants, then you can see that with the exception of this one, which I think is probably wrong, most of the information is lying here in the beginning of exon 11, where the VNTR region lies. So when you move even distal to this VNTR region where you retain the O-glycosylation sites, if you now have a frameshift mutation, it doesn't actually result in disease because the protein is then metabolized in the normal way. Okay, so then in conclusion, the F50 patient, which is homozygous for two loss of function variants in exon 2 of the cell gene, is in fact not affected, and those two variants are, well, the one variant is in fact benign, whereas the V670 patient, which is heterozygous, the variant results in protein misfolding and aggregation, and may be pathogenic. It's not necessarily pathogenic, but may be pathogenic. And I'm saying may because this is a novel variant, and in fact, it is distal to the most distal variant that has been described in the VNTR region up to date that is actually pathogenic. There are variants that's more distal, but they are benign. So this variant is lying between the pathogenic and the benign variants, and so additional functional studies is required to actually determine the pathogenicity of the variant. Okay, so some other conclusions, which I think is very important. Variant interpretation is sometimes very complex and very time-consuming. And perhaps very complex is perhaps a bit of an overstatement. I don't actually think it's that complex, but it's time-consuming in the fact that sometimes you need a lot of background knowledge to actually get to a correct interpretation. Sometimes it requires self-upskilling to mini expert in the gene or disease. So like I said, I didn't know anything about the cell gene, so you had to do quite a bit of reading on that. And even when you do that, I think the risk of error is still substantial. I'm not saying that I classified this very incorrectly. I'm giving a proposal of what I think it could be. What would be tremendously helpful is if there were focused expertise established in the country that deals with particular genes and the associated clinical conditions. So if there is established expertise in let's say acute and chronic pancreatitis that also includes the monogenetic causes of those clinical presentations, then that would be extremely helpful, because then would be background knowledge about these genes and the complexity that one encounter with these genes. Some other genes that we deal with quite regularly, like for example GBA, the gene that's affected in Gaucher disease, is also quite complex, but because we deal with it quite frequently, the interpretation is less problematic than dealing with something that you've never where you don't have any experience with. And then what I want to conclude lastly about this particular case study is that I think we need to think differently about our resources and the way in which we do things in South Africa if we want to make progress within the rare disease space. And that brings us to the Ingwe initiative, which is really our response to what we have experienced with the sequencing and the undiagnosed diseases work that we've done up to now. So, at the end of the day,
Chris,
yes.
I see there's 2 questions. I don't know if you want to take those questions now before you continue.
Yeah, maybe it's a good
idea. There's
idea.
a hand up. Yeah, because these are particularly about that case study. Jaco, I don't know if I can unmute you and if you want to discuss that.
Yeah, sure. Okay, so what Jaco is saying is not sure. Variant loss of function score moderate, indicating reduced penetrance, although dominant inheritance, and V670 located in the region predicted to escape NMD. So it was good that you were not sure about that, Jaco. I think I've perhaps answered your question in an indirect way. Okay, so there's another question.
Yeah, Jaco says I'm on my TV.
Okay, no microphone. But I hope I answered the question.
Yes.
There's another question here.
So there's another raised hand.
Yes.
Mitoli, looks like Mitoli. So you can speak. I've allowed you to speak if you want to. I don't know if your hand is raised by mistake. Okay. Seems like.
Okay.
Okay. Let's continue.
Okay, yeah, let's continue. So, okay. But I'm happy to take some more questions on that in the end as well. So the important thing here is that there are 350 million patients in the world affected by rare diseases. Of course, our interest in inherited metabolic diseases, but that's a subcategory of the rare diseases that's out there. And in any country, rare diseases as a whole is probably one of the top 5 health priorities. And I want to make a prediction that in the next 10 years, rare diseases will feature as one of the top 10 health priorities in all countries. It'll probably be higher than the top 10, but at least it will feature just as more and more information comes to the forefront and as more and more people realize that these diseases are actually very, very frequent when you consider them together. Now, if you consider our patient population, our country, and our continent, then you can imagine for yourself that there are literally thousands of patients out there that are going undiagnosed, never gets a diagnosis, never gets an opportunity for treatment, left with all sorts of stigmata and so on. And I think any one moral person that has witnessed this is compelled to act. And Ingwe is our response to what we have witnessed and the direction that we want to see this move in. So when I do presentations on inherited metabolic diseases, I often try very hard to convince the clinician that, even though he's only going to encounter this 1 in 100 patients or 1 in 300 or maybe 1 in 1,000, it remains important to diagnose that one patient, because you never know, the importance of that one patient. And this is the story of the starfish, of the girl throwing the starfish back into the ocean, and the man approaching her and telling her that she can't make a difference. And she responds that she made a difference to the one that she just tossed back. But this isn't actually the Ungwe approach. In the Ungwe version of the story, the girl, who is me, ask the man who is you, to go back and to go and get all of his or her colleagues, and to get their colleagues, and to come back to the beach. And then with fairly little effort, we can probably mobilize all of these starfish and get them back into the ocean. And that really is the Ungwe way. So in order to do this, what you need is not a principal investigator, but you need certain principles that will guide people's behavior. And in February, when we launched this, we came up with the Ungwe Pledge. And essentially what the Ungwe Pledge says is that, we who participate in the network will prioritize the interest of the patient above all other interests, that we will share everything, our expertise, our knowledge, our data, everything, that we will promote inclusivity, include our colleagues, and that we will respect the information that was generated from our colleagues. And it started off very humbly with only me and Ilse taking the pledge, and there's now almost 50 scientists and clinicians that has already taken the pledge, and this is very encouraging. So at the moment, and probably for the foreseeable future, essentially this is what Ungwe is going to come down to. So the Ungwe is open to patients both in the public and private sector, as well as hopefully soon to African patients. But the entry criteria is a little bit different. And what it comes down to is that you must have utilized all the resources at your disposal, reasonably utilized the resources at your disposal before you can be considered. So unlike our previous project, there's proper clinical phenotyping required, and then there needs to be an expert evaluation. So for example, if you've got a patient with a neurological presentation and it's a pediatric patient, then there needs to be a number of pediatric neurologists that would then evaluate the case and decide whether the case do actually is suitable for admission to the Ungwe program, or not, or are there additional testing that needs to be done before such a decision can be made. At the moment, the clinical experts do not exist just yet. So at the moment, it's pretty much just me and the clinician that discuss the case. But hopefully soon, we're going to have these expert committees that can do the necessary evaluation. And then once the patient is admitted to the program, pretty comprehensive analysis will be done. Excuse me. At the moment, as fast as we can fill up flow cells, we will do whole exome sequencing, and then within the next few months, whole genome sequencing, proteomics, metabolomics, and then also functional studies if required. Once a diagnosis is made, the patients are initially recruited to the Rare Diseases Biobank, and once the diagnosis is made, the information is shared with international registries, and the idea is to get the patients onto clinical trials or compassionate programs. Now, the important thing about Ungwe is that all of the data, as it is generated, currently it's stated that it has to be uploaded within a month. But I'd like to decrease that even further. So all of the data, the clinical data, the biochemical data, the genetics data, everything gets uploaded. And that data gets shared with all the Ungwe participants. Okay, so if you want to have access to that data, you need to sign the Ungwe Pledge. There are some more documentation that's on the way. Of course, it cannot just rely on a pledge. There's also a data sharing policy, legal agreement, and then ethics approval and so on. But these are things that we are going to share with you. The idea is to give you access to the data. Now, why is this important? I've just shown you a case of variant interpretation that, to be honest, I think took me the most part of a day to do. And as I'm sitting here doing this presentation, there are sequences and other biochemical results that awaits interpretation. And of course, if we can involve more people, train them and involve them in the interpretation, then of course we can get a diagnosis to a patient a lot quicker. And if you start to think about the number of patients that we're facing, then this really is the only way to do it. There's no ways in which any institution in South Africa can deal with the load of data that awaits us when we start to analyze these patients. Almost all of the analysis, or at least whenever it makes sense, is done in South Africa. We're not reinventing the wheel. Most of the stuff already exists. So, sequencing can be done in South Africa, whole exome sequencing, whole genome sequencing, proteomics, metabolomics, numerous functional studies, large immunological panels can be done. And the idea is really to approach people and ask them to make their resources available. Now, of course, I know that not everything can be done for free. And we will negotiate how that can be done. Keep in mind that, like I said, the UNGWE is not about a project and a principal investigator. It's about guiding principles. So all of the participants in UNGWE can approach for various institutions for funding on their own. Have their own patient set, submit their own data, share it with the network, and the network assist in them with getting a diagnosis to the patient. What we don't want is samples to leave the country. So, even though it may be slightly more costly to do in South Africa, we are going to continue to do it here. But there may be some analysis that just doesn't make sense to do here, and those we will outsource internationally. We've done extensive stakeholder mapping. I'm not going to go into so much detail into this, but essentially what it comes down to is when we deal with rare diseases, even though we're a diagnostic laboratory, we do realize that it's much wider than that, and there are many stakeholders involved and many priorities, and all of these have to be dealt with, and we are addressing this in various ways. If you are a young researcher or a young clinician, then you should take notice of this. So I just did a search in PubMed where I used the terms rare diseases, tuberculosis, and HIV. And of course, if you use different terms, you would get different number of hits. But the important thing is if you keep these terms constant, you can see that relative to tuberculosis and HIV, the number of rare disease publications have doubled. The percentage of drugs registered with the FDA that are rare disease drugs at the moment is 40%. So this field is literally exploding. And if you're a young researcher, young scientist, or young clinician, I do encourage you to get involved in rare diseases. And the UNGWE network is going to make that possible for you. Even though if you've got no sequences, no proteomics, no metabolomics, you will have access to the data, and you will be able to work up your patients, and you will be able to get to a diagnosis if a diagnosis is possible. So sometimes after this, I still get this question, what's in it for me? Okay, so I'll tell you what's in it for you. So the first thing is it's an opportunity to give back through sharing. What are you going to share? You're going to share your knowledge, your expertise, your resources, your infrastructure, your case studies, your data, possibly even your funding. You're going to share some of your time, even if just a little. So if all of us just donates a little bit of our time, we can move all of those starfish back into the ocean. You're going to have fantastic opportunities to collaborate with colleagues. You're going to be exposed to very interesting patients, specific cohorts, research opportunities, publications. We already have more data than what we can publish, so we're very happy to involve people in research and publications. You have an opportunity to mentor or be mentored. You will have access to a network that never rests. So hopefully in a year's time, when I do this presentation, somebody else is looking at variants that requires interpretation. And the other important thing is if we share this interpretation, we only need to do it once. If we interpret a variant once, then that information can be available to everyone in South Africa. It doesn't make sense for each institution to do the variant interpretation on their own. It makes a lot more sense to do that as a collective and then to share the information. You're going to get access to a network never rests, opportunity to get a diagnosis for your patient, access to experts, genomics, proteomics, metabolomics, bioinformatics, software, amazing training opportunities that's coming soon. And the most important thing is you're going to get the opportunity to escape monopoly. So I think, I know there are also private clinicians on this presentation, but in the public sector, I think we tend to play monopoly. We're all competing for Elof Street or whatever it's called these days. But there's a whole world outside of monopoly. And Unwe is going to give you the opportunity to stop playing Monopoly, to enter the real world and to actually make a difference. In the Monopoly world, it's about, who gets the publication and whose student gets a degree. In the real world, it's about getting a diagnosis to a patient and hopefully also to get a treatment to that patient. So in terms of what's currently happening in Unwe since we launched it in February, I think the most important thing to know at this stage is that we are recruiting. We are currently recruiting almost a patient a day. We're standing still a little bit now for the last week or so just because of Easter and some bottlenecks that we experience in the lab, but we are open to recruitment. You're welcome to contact me if you've got a patient that you think would benefit from such a workup. And I think the other thing that is very, very important is the training program. So we will launch that fairly soon, and the training program is only available to Unwe participants. So make sure that you take the Unwe pledge. But by taking the pledge, you become a participant, and then we will start to share everything with you. So with that, I want to conclude. If you have any questions, you're welcome to mail me or you're welcome to give me a call. And the one thing that you must absolutely do after this presentation is go to the website. It's also one of the things that we still need to optimize, but it is working. Just type into your URL unwe.org.za. You will get to the website, take the pledge, and we will be very happy to have you as a collaborator on the network. And so with that, I'm going to conclude, and I'm happy to take questions, and I see there are already a few.
Yes, you can just use the Q&A chat function if you want to ask a question, or if you want to ask it directly, you can just put up your hand and I will unmute you. So that in the chat, the question was just asked if we also do mitochondrial DNA. Is that included in Unwe? So I just answered yes, it is.
Yes. Yeah, I see Bertram Henderson has pointed out metabolic disease that is inherited in an autosomal dominant way. So thank you for that.
Any other questions?
While people are thinking of questions to ask, I just want to make sure that people understand the full scope of this. So we have at the moment limited funding to work up patients. But the idea is not that we have a project and that you join our project. The idea is that we create an infrastructure and a workway for South Africa that will enable multiple researchers and clinicians to run projects, provided that they share. That's the only requirement here. So, if you want to write an application and you want to use Unwe to strengthen that application, you're welcome to do so. So there can be research that can happen totally independent from us, as long as you just subscribe to the way in which Unwe works, which is really just a way of sharing the data. If you upload your samples, if you can't make a diagnosis in a month, give somebody else a chance. If they do make the diagnosis, then obviously you need to recognize them, include them in the publication. We will build all sorts of ways, we'll indicators into the database that will indicate who needs to be included into research and publications and so on, and also to make sure that people don't steal other people's data. I don't actually think that happens to any large degree, but we will also make sure that we'll take the necessary action to do what we can to make sure that that doesn't happen, and even if it does happen, that we have an audit trail to go back to see how that occurred. But it's almost like a network that can consist of cells that can pretty much function independently. And I think that's an important point to consider. We're also currently looking at establishing Unwe as a NPO, which would then, I think it's just important from a political perspective. It's not associated with any particular institution. It is its own institution, and essentially it provides you with the tools that you require to diagnose rare disease patients.
I see there's another question. I actually had a comment on that, Brenda. Brenda is asking what is included in the training program. So my comment was, if you have anything that you want the training program to consist of. So if there's any topic that you would want us to add to the training program, because for now the training program isn't set in stone, and it can be a lot of different things. There will be hands-on training, there will be diagnostic training, there will be scientific training, metabolomics, genomics. So there will be a whole, a lot of training that's very diverse. But if you have any ideas for training, you can please send that through to us. You can use on the UNWE website, there's a contact us form where you can add your comments. You can send an email. So Brenda, to your comment on what is in the training program, it's not at all set in stone yet. And we would love if you can send us your ideas. Then also I want to ask, if you can, please follow the LinkedIn page of UNWE and also the National Metabolomics Platform, because we will post all the opportunities for training there. We will also send these opportunities to everybody that has pledged, because that's the only way that we can have your information and your contact details. Is if you take the pledge, we will have your email and we can send you these opportunities. But I think the most important thing is to just follow this on LinkedIn, and then you get more information. Brenda, I hope that answered your question for now.
Yeah, if I can also just add a little bit to that. So, I mean, the first data that is going to be available on the central database is going to be exome data. So, one of the highest priorities is at this stage is to give people training in the interpretation of exomes. And it may sound like a daunting task, but there are some pretty clever software out there. Part of this I've shown you, and I can assure you that within a weekend, you can probably start to do variant interpretation. I'm not saying that you're going to get everything right, but you can start, and we will build in various checks into the process to ensure that if you do get it wrong, that somebody else can correct you, and that the necessary training can also take place. So that is a very high priority. But like Ilse said, we're also open to suggestions.
Yes, and the idea is also that these labs do exist already. You mentioned this in your previous talk as well. The labs are there, the expertise in the labs are there, but we can provide training to those people who want to sit at home and interpret the data. So the training is, you don't necessarily have to understand how the sequencing in the lab work or how the metabolomics in the lab work, but we can provide training so that you can look at your own data and understand what you're looking at and mine your own data. Because there's two more questions. So in the chat, Magda is saying, Chris, we need to include the genetics expert committee of the NHLS on board. I will contact you to schedule a meeting. Okay, that's in the chat.
See, Frank Lewis got a question. Is there a pathway to test for repeat disorders, e.g. expansions by means of repeat prime PCR or contractions by southern blotting or optical genome mapping? You're talking about tandem repeat expansions like Huntington's. Maybe we can unmute Frank Lou, Ilse.
Yes. Okay, Frank Lou, you're allowed to speak now. You just have to unmute yourself.
Okay, but I mean, maybe I hope I'm going to answer the question. So whole exome sequencing is not always the best technique to use for tandem repeats, and often it's prone to errors. But the whole genome sequencing that follows will be able to pick up those repeats. And then, of course, there are also various other targeted assays that one could do. And some of these assays are already established in South Africa. So, where there are assays that we can utilize, we'd rather utilize those assays than to, come up with a very reinvent the wheel, very expensive technology that we can use. So, if you do have some assays that you would like to share to the network, then, we're also looking at ways in which that can happen.
Am I unmuted?
Yes.
Hi, Chris. Yeah. So look, even whole genome sequencing will not pick up the repeat expansions that we see in neurology, like myotonic dystrophy, Huntington's, and so forth. And the bigger problem even here are the contractions. FSHD or facioscapulohumeral dystrophy is Definitely one of the most common muscle disorders, inherited muscle disorders that we see. And for that, southern blotting at the moment is a gold standard. Optical genome mapping is making headway, but it's impossible to pick those up by either whole exome or whole genome sequencing.
I'm not so sure about the whole genome, but it's something that we can look at. But again, come back to what you just said. If you've already got a technique established for that, then we'd rather utilize that than to now come up with something new. I mean, one of the things is to put all of the resources in one basket and to make it available to people so that they can start to use it.
But that's exactly my question. There is nobody in South Africa that's doing southern blotting for FSHD, for example.
Yeah. Okay. So then there may be an opportunity there that we should look at. I mean, obviously there is going to be a lot of functional testing and other testing that we don't necessarily have access to. If it's a very rare occurrence, then the approach is still to refer it internationally. But like you say, if there are a number of patients that can benefit from that, then it needs to be set up. And it sounds like you guys already have the expertise. Well, the clinical expertise. It makes sense actually just to provide you with the lab expertise to set it up there, and then people can refer to you. So try and to pool all of the expertise into to make it available to the country, not to pull it all into one place. Use the expertise wherever it is established. I'm not aware, except for Huntington's, I'm not aware of, or I'm not that clued up with any other expansion repeats and who test for them. Huntington's can be tested by the private paths. Yeah.
Myotonic dystrophy as well.
Okay.
But there are some others that's not being done in South Africa. Canvas, for example.
Okay. Yeah.
But anyway,
so I
it's a
mean,
long
yeah.
discussion.
Yes. Tumelo is saying this will provide some great learning opportunities in the growing field of omics. Quite exciting. Yes, that is exactly what we want to do. And I think, the important thing is we want to reach the next generation of clinicians and scientists. And with next generation, I think, anything of, 40 or, I have to be careful now. Yeah, if you're going to be in practice for the next 10 years, you should give us some thought, because the technology is expanding very, very rapidly. And if we can succeed in establishing such a network, where we can utilize everyone as opposed to everybody trying to do their own thing in their own ivory tower, we can make some serious headway. We can go from, I think, where we are at the moment, which is very far behind the rest of the world, to actually move up right to not to the top position maybe, but certainly to the front end of the pack. And yeah, all that's required is for people to put aside the other interests that's often competing, the institutional interests, the research interests, the publications, all of those things. Put the patient first, and then we're going to make some serious headway.
There was a question sent to me personally that I can just ask. I see we have some time left. So the question is for the audience members. So this isn't for you, Chris, but for the audience members, what are your expectations from NGWE? What do you expect from this? And what is your concerns with a program like this? And this question is from Elna Conradi, who is the biobank head at the CHM. So we know that people have a lot of fears when it comes to new things. So she just wants to know if there's anyone who wants to answer. You can just put up your hand. And if there's no fears.
Look, I mean, I have some fears that I will mention. So I mean, of course there are ethics concerns here. So we're talking about large-scale sharing of genomic data, and there's a lot of bad things associated with that. And currently we are looking at that, both getting legal as well as ethical opinions, but of course there's also bioinformatics that I think can contribute substantially. So one way in which we're looking at this is to give people read access to all of the data. So if you want to interpret, you can look at the data and you can interpret, but you can't actually download anything, and you can't see the raw data. You can get access to the raw data, but then it's a research project, right? Then you're not doing diagnostics. And then, of course, you need to have a research protocol and so on. And so we're looking at, how we can incorporate these things in such a way that we enable people to get broad access without compromising on patient confidentiality and these things. So I think anybody that hears this must have some concerns in the back of their minds. And if you do have some and you have ideas, then I welcome you. Well, I invite you to contact me and to assist with establishing these the setup in such a way that we lower the risks as much as we can.
Yeah, so there's no other questions that I can see, but as Chris said, if you have any other questions, please feel free to send an email, go to the NUE website, send us through the contact details. You can also go to the National Metabolomics Platform website and use those contact us channels. So this was the first presentation of a 3-part series. So next week, same place and same time, we will have Dr. Engela, let me just say yes, Dr. Engela Honey, and she will present a case study on patients with UNC80 deficiency, and these were patients diagnosed through the NUE program. And then in 2 weeks' time from now, we'll have Prof Chris Hendricks, who will talk about the value of a collaborative clinical approach and rare disease education. So please join us again next week. Thank you very much, Chris. That was very insightful, great talk, even for me who doesn't understand a lot of genetics. That was very interesting. Thank you everyone for taking time this time of the day to join us. We really appreciate it. Yes, I don't know, Chris, if you have any last words. Otherwise, I think we can close the session for tonight and you can all have a good evening. There's just
no last words. Thank you to everyone.
We have a thank you from next week's speaker. We'll see you then. Okay, enjoy your night.
Thank you.
Thank you, everyone. Bye-bye.